One of the points I made this morning in the lecture was that in pediatrics, we've learned a lot about how the body gets rid of drugs. We're learning more and more about the importance of genetics and genomics. But the one area, and arguably the most important area for pediatricians, is how do drugs work? What do they do? We have done a bad job with pharmacodynamics for many, many years. And so our group at Kansas City is working diligently to try to come up with ways, child-friendly ways, to look at this. So today's lecture, that's what we're going to focus on. I have this brand new technology here. And I'm told if I do this, that happens. They said, only touch the screen if you want to move your slide. So I'm going to step away from the screen. So this is just a model. But, hello? I'll actually, on occasion, a lecture, if the student phone goes off, I'll pick it up and answer the call. It's the funniest thing in the world. So pharmacologists, one of the ways, one of our tricks is to make models of things. And this is a model. I like this picture because it kind of wraps it up in one picture. Drugs are used to treat diseases. They're prescribed because we expect they're going to work and do something beneficial. And at the end of the day, there's some relationship between that drug in the body, wherever it goes, and how it works and for how long. So mathematically, we can make models of this stuff, and people do. But when we talk about pharmacodynamics, it really is that relationship between that small molecule or maybe a large molecule and some bit of biology that produces an abnormal condition. So to be able to measure drug effect reliably, especially in kids, is huge. And it's something that needs to be looked. So, biomarkers. Really, if you think about measuring drug effect in kids, almost everything we do is incorrect. We have to rely on biomarkers. And this is a definition of a biomarker. I like this definition very much because it says it may be used to see how well the body responds to a treatment, a disease or condition. This is the official NIH definition. And I'll bet the Canadian version of the NIH has their own definition. Everybody's turned it around a little bit. Finds it as a characteristic. And it's broad and actually includes pharmacologic response. So it's okay to talk about measuring pharmacodynamics in the context of a biomarker. Now this is a table, and this is not inclusive, but I use it to illustrate some pediatric biomarkers that are out there. Many of you will know these. I've put them in three categories. There are a group of things that we use to look at disease expression or response. Some that deal with systemic exposure, so a lot of the genotyping assays, so TPMT for instance, and azethylamine. That gives us some idea if somebody can't metabolize it because of defective activity, the exposure is going to be there. And then some that really measure effect. I always challenge our fellows to go back and read the clinical pharmacology literature of the 1960s. To go to the library and get an issue of Journal of Clinical Pharmacology or Clinical Pharmacology Therapeutics and see how many of the papers in there dealt with hypertension, drugs to treat hypertension. Why? Because you can easily measure blood pressure. So many of those you know. Here's an example of biomarkers, CD45 cells in marrow for disease progression monitoring. The bone marrow transplanters use things like this all the time. You know, when they've got a graft book, it works. So these are very, very useful. You can look at them repeatedly over time and see them move in patterns that may be time dependent or treatment dependent. It's one of the reasons it makes them so exciting. Well, we're talking about pharmacology, so I'll go back to the exposure-response relationship. And there's a whole area in the pharmacology world where people have taken these concentration effect curves, and we can generate parameters that describe quantitatively things about the effect, the EC50, so the concentration where you might see half of the maximal response. Go back into the early 1980s and look at the studies that were published on the pharmacology of antipyretic drugs in children. Again, it's easy to measure body temperature. So a lot of the application of this stuff was applied to that, which is where today we have our doses of acetaminophen and ibuprofen used to treat fever. It's all rooted in things that were pretty descriptive that we could get at. I showed these data this morning. I showed them again because I really think they're key. But here, there's two proton pump inhibitors, pantoprazole and omeprazole. See, I'm pointing it out, not touching. Elimination rate, constant area under the curve, a relationship with pantoprazole. But down here are the genotypes, the 2C19 genotypes. So for pantoprazole, you can actually take those genotypes and turn them into a predictive activity score. So you can go from genotype to predicting the plasma concentration of pantoprazole because it's such a good marker for a particular people. So it's another example of a biomarker. And it's really, I mean, that's a linear relationship. That was exciting. Those were data that my friend Steve Leader, initially he said, we don't need to look at those samples again. I said, oh, there's something. Well, pantoprazole, what if we wanted to use it as a biomarker in kids? Can we give kids a dose of this stuff? Yeah, used routinely. But what if we could label it? Okay. What if we could label it with C13, which makes it not radioactive? What could we find out about its biotransformation? And the answer is not. And this could be given to kids. In fact, Jay Lee, we're going to do this next month. And why are we going to do it? This is a cartoon about clopidogrel. It's a drug used mainly to treat adults, but it's pretty well substantiated that knowledge of the 2C19 genotype is predictive with regard to people who are going to respond to clopidogrel. And if you had the ability to take C13 pantoprazole and make it into a breath test, okay? So the breakdown of the molecule happens, the label comes off, it's transferred, comes out in expired air. You ought to be able to have a non-invasive way to learn some things about the drug. And indeed, these investigators have shown that. Pantoprazole breath test. Okay. Same technology that's used to diagnose H. pylori. You just hook it to a drug that in this case is a substrate for a specific enzyme. I mentioned this morning a quote from Nick Holford. Nick is and pardon me, I have to walk back. I've got these progressive lenses in my glasses. It's part of growing older, so I have to get to the point where the focal length of my lens screw this up. So this is what Nick says, the first quote. Time to put into practice. If we're going to do studies of drugs in kids, we need to not only be focused on what happens to the molecule and all that stuff, that's all well and good, it's important. We need to understand something about how it works. He went on to say that pharmacokinetic studies without pharmacodynamic studies are unethical. I don't believe that. I think that's an overstatement. I've told Nick that. And in terms that we're actually a little more harsh with that. Wouldn't it? Yeah. But he's a good opinion. So the challenge in kids of doing this stuff is that if you think about where drugs act, they act in the biophase. They act, and this is another model depiction, but they act at a place, that receptor is in somewhere that generally is so distal to where you can sample, it becomes impossible many times to directly assess the effect. Okay? But just like walking on the sand on the beach, if you turn around and look at your footprints, you can know something about where it was that you were. And the same is true of this. So coming up with approaches that may not be down here, but in areas that we can access and measure, gives us a chance to know something about drug effect. But there are challenges. For sure there are challenges. We went through some of these this morning. Lack of validated endpoints. Okay? If you go to a regulatory agency and you start to say, I'm going to put a biomarker in my clinical trial, some regulator is going to look at you across the table and say, has it been validated? Okay? And if it's not validated, then in a regulatory context, the information may not be considered to be admissible, which is a problem. In adults, we can do all kinds of things. We can line them up outside for-profit CROs. We can pay them $1,000 to do muscle biopsies, repeated endoscopies. Okay? In the US, this happens in the, I don't know if it happens here in Canada, in the US it happens. You know, you see advertisements in the paper. And people, some of whom are professional study subjects, sign up and get poked and prodded and all manner of things. But in a child, you can't do this. You shouldn't do any of it. Okay? And I told the story this morning, the proton pump inhibitors. The FDA used as their endpoint for proton pump inhibitor studies in kids. So they were trying to get these drugs labeled for use in kids because they were so much used. Their endpoint was histologic evidence that gastroesophageal reflux disease had improved. How do you get that? You stick a tube down there and you take a piece of tissue. Couldn't do studies in pediatrics. And for a decade, the FDA didn't budge. They didn't budge. They watched these drugs be used. A couple of them were, the last year that omeprazole was a prescription drug in the United States, when it got 6 months of additional exclusivity, it sold $4.4 billion. They got 6 months, they got another $2.2 billion. They didn't have to advertise that drug. They're huge, some of these things. So endpoints become critical for a company when they want to label one of these drugs for kids. Pediatrics, acute diseases predominate and oftentimes fluctuate in a rapid fashion. You kids, they're tougher than we are as adults. They get better and they change quick, which is a good thing. But sometimes that muddies up the water when you're trying to do pharmacodynamics. And then the potential for development to influence drug receptor interactions. There are cases where that's very, very real. Beta receptors are expressed differently. Probably the calcineurin pathway is a bit different in terms of pain. And here's some data dealing with the calcineurin pathway. I showed it this morning. Cyclosporin, we did this in 1999. And using some in vitro techniques and cells that were obtained from kids going on cardiopulmonary bypass. Lots of blood available there, you know, easy to get to. And we looked at cyclosporin using a very traditional approach in pharmacology and found that little kids required half the amount that older kids did. Now, we don't know why this is, but it truly was something that was part of development. So, it's 2013. We've got all these academic leaders saying we've got to do pharmacodynamic studies in kids. We have challenges. How do you do it? There's got to be a better mousetrap, right? Got to be a better mousetrap. Or I would argue that that mousetrap is fine, and that's a better mouse. So figured out how to ease this way onto the cheese. That's how I think of our research sometimes at our place. We're easing our way onto the cheese. Okay, let's do some examples. Any of you ever had one of these? Happy spitters, they're called. These are the kids that get put on proton pump inhibitors, metoclopramide, H2 blockers, all manner of things which our gastroenterologist in the audience knows, for the most part, don't do anything except make the mother feel better. The kid still vomits. And they're happy. So here's metoclopramide. Now, I was fortunate. We did some of the early work with this drug in pediatrics. We have no prokinetic agents really today in pediatrics except this stuff. It's messy. Newborn babies, about 20% of kids in the NICU will have some CNS adverse effect of this medicine if you give it to them. Okay? They don't feed well, they don't do well. And if you look for evidence of whether it works or not, you really don't find much to say that it is a very good drug to treat reflux. You find a lot that says this thing has some severe adverse effects. But how much metoclopramide do you think is prescribed in North America? Children. A lot. A lot. Okay. Even family physicians will give it out because there's rarely an option. So attempts to study prokinetic drugs in kids over the years have really been problematic, in part because we had no easy, child-friendly way to measure whether these drugs were doing their job. It's affecting motility. We didn't have non-invasive approaches, and all we had were things like manometry and then dye tests and pH monitoring. And if you think about esophageal pH monitoring as a motility test, it just says that what should go south went north. That's all. And that's not good. So it's really kind of crap shots. Lack of specificity and sensitivity. Again, esophageal pH. That was the gold standard that the FDA used to look at prokinetic. And then what about drugs with different mechanisms of action? We were given acid-modifying drugs, and the FDA wanted to do esophageal pH monitoring. Okay? Well, that tells you something about the acid that's reflux, but it may not tell you anything about the disorder. So, yeah, problems. Then if you want to study this, let's say you want to study gastric emptying. How do you really study gastric emptying in a kid? You give them some scrambled eggs or some infant formula with technetium. You schedule a block of very expensive time in the radiology suite. You try to get them to hold still while they go into the machine, and then you watch what happens. That wasn't the kind of test that would get you very far if you wanted to study a prokinetic drug. So we needed something better. So what we did is we went into the GI literature and we found applications for C13 labeled octanoic acid and C13 acetate. And people had looked at these things in adults as ways to do or examine gastric emptying. And it's pretty simple. You give the kid a dose of something and you collect exhaled breath. Why this kid decided to blow that up with his nose, I don't know. It's not my child, but that's something my child would have done. And so we did this in a group of kids, and we compared it to scintigraphy. And what was interesting, and I think the next panel has the data. Yes. Yeah, I'm going to have to go. I'm going to go back. But so scintigraphy is done in a certain way. You literally measure that disappearance half-life. Okay. So when we plotted our results versus the scintigraphy results, there was an association, but it wasn't just striking. You know, the R squared wasn't 0.9 at all. Let's see if I go back. Yeah, work. So we chose to look at it differently by saying, well, maybe it's just not the half-life. Maybe it's the cumulative area under the curve. And when we did that, we found better associations. Okay? Two very different tests, looking at something similar, but it was how you looked at the data that kind of brought it to life. So let's switch gears now. So C13 breath tests, we like them a lot, we're using them, and we're going to keep using them for doing things. We're doing a pantoprazole study this summer that looks at the question of obesity, intermediary metabolism, and it's a big metabolomic study. So we're all ready to go for that. We had to get an IDE to do the study. You know what an IDE is? For FDA? It's an investigational device exemption, because the FDA regulates it as a medical device. In all of my years, I've never filed an IDE. It took us about 3 times before we finally got it right. So we got it right. We have our IDE. A couple of years ago, at least in the lower 48, there was concern expressed about over-the-counter cough and cold medicines. And the fact is that these are billion-dollar product lines. They were used extensively. People self-medicate. I'm sure they do that in Canada. And there was precious little information about the study of any of these drugs or combinations of drugs in kids. And there was some sense that they were potentially doing more harm than good. And so in the absence of data, the FDA basically put the kibosh on them. Kids under 2, no. No more. And they made it very public. Okay? It went into force very quickly in the U.S. What do these things have in them? This. Diphenhydramine. Synthesized in 1941 by George Rivichel from the University of Cincinnati. First went on the market in 1944. It's been the largest selling antihistamine drug in the history of all antihistamines. Very old, commonly used in pediatrics, from everything for snotty nose to sedation, just lots of diphenhydramine still used. And one of those drugs that in young kids can produce paradoxical CNS excitation responses. So not necessarily what you desire. Histamine is a very interesting molecule. And for all the antihistamines that are used, studies of histamine and antihistamines hadn't been done for a couple decades until recently, when we really appreciate that it is a very, very important mediator in many, many places in the body. It's just not mast cells. It does lots of interesting biology. And it's everywhere. In the heart, in the head, in the nose, and the toes. Histamine is everywhere. Okay? So if antihistamines are a big drug class used in kids, and if we don't have data to understand their pharmacodynamics, and because it's such an important player, it stands to reason that maybe we should learn something from it, and not just take it for granted. So if you go into the histamine literature, the allergy literature, you can find the description and use of the epicutaneous histamine test as a way to look at antihistamine effects. And it's not a bad test. It measures wheal and flare. It's the gold standard. You can actually find a few pediatric antihistamine studies done through the years that use this test. But it's kind of a bear to do. There's a chance for the technique not to be done right to place it. There's a chance that people reading it will read it differently. While there are ways to digitize the image and be a little bit more exact, most of the time it's done by somebody with a ruler. So what might the data look like using this approach to try to critically study an antihistamine? Could it look messy? Yeah, it does look messy. So we started, again, we went back. Sometimes I think we're just borrowers, right? People in pediatrics are borrowers. We look at what the adults do. And, for some years, the surgeons used laser Doppler monitoring to look at microvascular blood flow. Physiologists have been doing it for years. And so we thought, well, maybe this could be a pretty good tool if we were able to give a dose of histamine and measure the microvascular blood flow. It's pretty easy to do. It has distinct advantages over the epicutaneous test because it's far more objective. This is the machine hooked to an arm. You can see there are two little things. One of those chambers, I think I have a better schematic. There we go. Okay. One of those things is an iontophoresis chamber. So it uses a very low current to drive the drug through the skin into the microvasculature. And then kind of next door is a laser flow probe. And it's measuring, as the cells go through the microvascular, it's just measuring the movement in terms of how that scatters the return laser line. It's really a neat technique. It doesn't hurt, and people aren't bothered by it. And when you look at the output from it, this is an example of the output. So this is time, and these units are changes over baseline. Okay, changes over baseline. Now, it was interesting. So one of our fellows, who's an allergist, and was doing this work, and she started to get these curves back, and she came to me and she said, this looks really good, but I don't know how to handle these data. How do you analyze things? And I said, well, what does it look like to you? And she had trained with us as a clinical pharmacology fellow. She said, it looks like blood level data. I said, well, let's analyze it that way. That's what it is, right? What do you have an onset, a maximum, and then over time, a decrement. It could just as well be a drug concentration. So we had to make some new parameters and some new terms, but it all worked out for the better. And so we were able, and that output that I showed you, that's cleaned up. The output you get from this machine, in fact, the real story is it gives you a continuous reading, and it produced so many data points that when they took it to a modeling and simulation person, and they tried to simulate it all, it crashed the computer. So we had to come up with ways to come up with how to pick points that were, data rich and represented. But what Bridget has found is that if you look at this, and we validated it in adults, we did the whole story. But what she found in kids was when you looked at enough kids, you actually found evidence of 3 response phenotypes for histamine. Never been talked about. But for those of you in the room who are pediatricians, you give an antihistamine to one kid and it works. You give it to another one and it doesn't work. What do? You switch or you escalate the dose. So we've known, based on therapeutic use of these drugs, that there are response phenotypes. But with this technology, we're able to discriminate them. And actually this, Dan, I think is in JCP.
There's one coming out in July. I just approved it today.
Yeah, so we're putting this in the pharmacology literature because we think it's real pharmacology. But we're going to get it into the clinical pediatric literature very soon, coming to a theater near you. But this stuff is really, really neat. So if you think about it, now if we were going to study, let's say a company made a new antihistamine, a non-sedating, highly selective, potent antihistamine in a formulation that you could reliably give to kids, and you wanted to study the concentration-effect relationship, you would need to know which phenotype responder that kid was. Because if you didn't, you'd put them all in one group, right? So here we can separate out the groups. What causes the difference? I have to tell you, we don't yet know. Okay? It may be something as simple as the biotransformation of histamine. Okay? Diamine oxidase and histamine N-methyltransferase, we've looked at this a little bit in populations. They're potential candidates. And why? Because when you look at them, there are known polymorphisms, allelic variants of the gene that have functional consequences. So we are doing this now in a big population of patients to see if we can figure out what makes this phenotype. Okay, here's another story. I like this. Those look a lot alike, don't they?
Can I interrupt for a
Sure.
second? So the story on the cold medicine case, right? That was the previous one.
Yeah.
So it was taken off the market because these babies, they were hyper responders and responders are wrong.
Not really, no, not totally. It was taken off, they were taken off the market not to be sold to young kids because there was no evidence of their efficacy, because there were no studies, and some concern about their safety.
That was a very long standing.
Yeah.
I know in the Netherlands they took daptropine out in the 90s because of hallucinations and that type of reactions in young kids. That has been an ongoing problem
Yeah.
for a long time.
That's just no data because people hadn't studied it, and in part because they didn't have the right approaches that they could do it.
Well, they never had PD data to begin with.
No.
I mean, the original doses were just age-dependent, half an adult or quarter of an adult.
Yeah.
So they would, I sat on the Canadian committee that looked at this as well. We came up with our recommendation, which was a little more harsh. It was clear that the reason for lack of efficacy was that there were never any PK/PD studies. So whether these kids were being underdosed was probably the problem.
Yeah.
The other big issue is the abuse potential. I've had an unfortunate court experience with that kids know about the pharmacodynamics. and especially with diphenhydramine, the safety margin between efficacy and toxicity is very narrow, and there's been a lot of deaths that have caused a lot of court cases.
So I certainly, don't get me wrong,
No.
I certainly supported removing from the market, but the justification of lack of efficacy was simply because they don't know what dose to give.
Well, sure, I totally agree, but, and then maybe those cases of deaths had a response phenotype that put a certain group of kids at risk.
They were abusing it. Kids
Oh.
know that recreationally,
Oh, yeah.
big time.
Because of the bubble gum.
I use it to fly
No, it wasn't,
transatlantic.
it wasn't bubblegum flavored. These guys
I guarantee you, if you take this drug, you won't get high off of it. You'll get low, really low, if you don't take the right amount of it. Methotrexate's an old drug. Many of you know its story. It looks like folate. It makes the body believe that it could be folate, and that's the basis for how it works. This is a cartoon which talks a little bit about methotrexate and folate. The biology of this is very interesting because both folate and methotrexate become bioactivated by becoming polyglutamate in the cell. That's both the drug and the endogenous thing are polyglutamate. But we have a rheumatologist, Dr. Mara Becker, who did a Clinton Farm Fellowship with us, very bright woman, and she was troubled by what she saw in the clinic with her patients with juvenile idiopathic arthritis and the adverse responses to methotrexate, which they use a lot as a disease-modifying agent. So she began to work with Steve Leader and others in our group to look at biomarkers to see if there were any signatures that might be associated, in this case, with GI side effects. And she found, and even though there's lots of variability, it's biology, right? That's what happens. But when you looked at the total methotrexate polyglutamates, and then just the smaller ones, the 3 to 5, that they seem to be different with respect to GI side effects. Yes, no. There seems to be something there. She also found, and this has just recently been verified from another group, an apparent difference in the bioavailability of the drug that was predictable in part based on its polyglutamation status. But there was a difference between PO and sub. And again, no one's figured it out why. I have some hunches about the importance of something seeing the liver first. But a biomarker that's beginning to explain some of the challenges that she sees with the drug she uses a lot. Now let's move on to pain. This could be rather painful. How many of you have experience in assessing pain in a nonverbal child? It's not easy, is it? Nurses do it all the time. I still work on our clinical consult service 1 week a month. I don't know how I do it, but I have good fellows. All the time, you go to the nursing units, and I'm sure this only happens in Kansas City, but a child is put on an opiate, the baby gets restless, they're looking for sedation, and what do? Give more of the opiate. Okay, probably more misuse of opiate analgesics than we ever know. But they're important because they're the most effective pain treatments that we have. And all of the visual analog scales, all of the stuff that people use routinely to clinically assess pain can be useful, but they all have their foibles. They're not so easy. Well, one thing that opiates do is they change your pupil size. So Mark Conley and Kathleen Neville at our place, Jacob Brown, one of our fellows now, have been for a while looking at pupil response in children using a handheld pupilometer. It's a really cool device. You can get one from NeuroOptics. It's about $7,000. If you get two, they'll give them to you for 12. Quality of discounts. And these are just some raw data from a number of lines, there's a number of patients. But this is the maximal pupil response and the visual analog pain score. So Mark and Kathleen Neville found that this was really pretty predictive, so much so that when they looked at opiate dose, okay, opiate dose, and maximal pupil size, minimum change in pupil size, constriction velocity, dilation velocity, they found significant associations for all of those. So it seemed to be really real. To the point, there's 440 assessments, average constriction velocity. Now this is smooth, because we could fit this to a function, but we have. And morphine equivalents, they showed me this, and I said, there's something to this. Okay? So all of a sudden, think about if you could measure pupillary response and be able to turn that into the prediction of an opiate dose that you would need to treat pain, might that be useful? Yeah. Be very useful. Or better yet, a nomogram to calculate the exact dose. Now, they haven't validated this nomogram yet, it's in process, but very exciting stuff. So they showed this all to me. And Jacob Brown, one of our fellows, and said, well, what do you think of this? Am I okay on time? Okay, just if I need to. I like telling the stories. And I told Jacob, I said, well, does this pupil response change in children as they get older? Does ontogeny affect the response? Because if it does, then how do you know any of this really is what you say? So anyway, did the study. Presented it at ASCPT this year, and even though, against my recommendations, he drew lines through those dots, I can tell you that those relationships have no association, and it does not change as a function of age. And that's a good thing. Which means then they can continue their studies with this pupilometer in kids and use it as a way to look at the pharmacodynamics of opiates and the response to them. Okay, one more story. Infliximab. Pharmacologists now, we have to think about big molecules, not just small molecules anymore. That's its picture, that's what it weighs, it's pretty much distributed into the blood volume, intravascular volume. And if you look at data on a host of these things and people coming up with predictive equations, you see something in these that kind of 0.74, 0.75, that's a scaling factor. It's an allometric scaling factor. 0.75 is a scaling factor that some people believe that you can predict and translate stuff from a mouse to a human in terms of clearance of a drug. So I had a friend of mine from Johnson & Johnson who they conducted a study of infliximab in juvenile idiopathic arthritis. They were trying to get labeling. FDA denied them because they didn't meet their endpoint claim. And he said, would you look at our data? I think there's something funny. So I sent it to him. Here's plasma clearance of infliximab, 117 kids. Association with age. Looks like a lot of, it could be a small molecule. Higher clearance if you're younger. And so he said, why is that? He said, I can't explain it. He said, this drug's not really metabolized, and it's not. Monoclonal antibodies are pieces of proteins. Okay, they get recycled through a scavenge pathway, or they get tagged for proteolytic destruction. They get ubiquinated and broken apart. So it immediately didn't make sense in the context of what we know about kids. So I began to think about it. So well, what about this outside the salvage pathway? What happens to a protein? Well, it's all intermediary metabolism. It's all energy-dependent processes. That's the only way it gets chewed up. Well, what do we know in children about energy expenditure? These are data from the World Health Organization. Age, resting energy expenditure. Here I chose to normalize it because kids weigh different amounts. Highly correlated. Pediatricians have known this for eons. The younger kids, you say terrible twos, the kid runs around everywhere, got a bundle of energy. That's because they are a bundle of energy. They have to be. Think about it. What's growing? Organs are developing. Kids are getting taller. All kinds of stuff. Well, just what if that age-dependent increase in energy had something to do with the clearance of infliximab? There's the data. That could be a small molecule. Now, we published this. You'll notice we buried it in an obscure journal. So no one would find it. And so a lot of what we see in development might be nothing more. Think about how P450 works. It's a hemoprotein. 1 milligram of P450 in a 2-year-old and 1 milligram in a 22-year-old ex vivo substrate, same genotype, completely catalytically active. Put that P450 in a child's body, an adult's body, and it's a whole different story. So stay tuned. This is part of our summer fun that we have coming up. And we've known this for years. Here's phenytoin, Vmax for phenytoin, much higher in little kids than older kids. And it's true, it happens. We need to find out why. So, where's the future going? Pharmacodynamics. Many people think here. We have this power now to bust through an exome in an afternoon. Find out many, many interesting things. I remind you, there's a whole lot of junk in the human genome. And it's separating out the gold from the junk that will be the challenge. But there'll be more to come. Now, I mentioned should biomarkers be validated? Yes. And there's a process. We're not going to go through this. But there is a process. And I would challenge any of us who might want to work with one of these to keep this in mind. You might not be able to go through the whole regulatory piece of it, but in terms of scientifically bringing a biomarker forward, if you pay attention in your studies to this process and do as much of it as you can, at the end of the day, you'll make a bigger contribution. Because when you look at drug development and where you can put biomarkers, anywhere in the chain. There's a place for them from early preclinical all the way through post-market. But it's not reductionist in need. Okay. This is a reductionist in need expression of a couple pathways important for cancer. Bob Marcisi reminded me after he showed me this slide that this is what it really looks like. Okay. This is what the human genome is giving us information on. Figuring how these things relate to each other is going to be the holy grail of it all. So, what do we have to consider when we look at these? Growth and development. What's the proximity to what we're measuring to where the drug works? Is it dynamic? Can we look at it over time? Is it acceptable? Is it child-friendly? And is it accurate? Is it specific? Sensitive? Is it an accurate way to do it? So, conclusion, we need to look at PD. It's going to require us to make biomarkers. In some cases, proof of concept can be accomplished in adults. In other cases, not. So, a success in doing this is going to require collaboration between people who understand the molecule who understand the physiology, and who understand the disease. Teams of people make sense of this stuff. Very few of us can do it in isolation. So I want to thank you for your attention, and I think I'll stop there.
Thank you very much. He also referred himself to the chief storytelling officer that Joan Cosby comes from, and you can just tell from the round, it's a beautiful scientific story told in a wonderful way. But we do have some time for questions, probably 3 or 4 questions. So who would like to?
In your morphine and your pupil size study, were you able to incorporate the ontogeny of the 6-glucuronidation pathway that complicates the assessment of analgesic response?
So far, they haven't put this in a pharmacokinetic dimension yet. Okay, so all of the work that that group has been doing has been to validate that pupilometer and to get ready to be sure. The last question we had to answer was the age question, because I was very concerned about that. Why? So morphine glucuronidation is a great example. 2 UGTs do it. Both of them highly polymorphically expressed. One of them with a frequency that's significant for both Caucasian and African origin populations. So if you launched into a study and you're using this pupilometer and you've got some age-effective pupil response and you didn't know it, and meanwhile you're looking at all these metabolites of morphine and you're doing the genotyping and you're trying to make sense of it all, it could have been a big mess. So that's why I said stop, do the normative study. And that's what they're finished, they're writing up now. Yeah.
Continuing on that theme, the challenge, as you know, in trying to decide about opiate therapy in the preverbal child is when they're agitated. And are they agitated because of underdosing of the opiate, or are they agitated because of adverse effect? If it were an adverse effect, there's the likelihood of the sympathomimetic pathway, which would, of course, give the opposite result to the pupil. So in those studies, have they been looking at whether or not there may be that component present? In other words, is the pupilometer validated in issues where we're dealing with an opiate toxic rather than a subtherapeutic?
They've not done that yet. The studies that were done, again, focusing on trying to develop the tool, were done all in elective post-op surgery kids. The biggest cohort was kids who had a pectus repair. So that is admittedly more reductionist and neat than the kid who comes out of the ICU and can be thrashing about for no other reason except they don't know what night and day is for 10 days. It has nothing to do with the drug. But I think you really have to know your tool before you apply it. And so this group has, I told Jacob Brown, you have a whole career in front of you with this if you want to take it.
Because certainly there's sympathomimetic drive going on, that could give, certainly affect the pupil size.
Yeah. The nice thing about the pupilometer is size doesn't matter as much as response.
Could take that competition in other directions.
No, that's not it.
It could be a Cialis commercial. They play that on Canadian TV.
In Canada, we don't.
It's the same nauseating commercials.
You know,
You gotta work the channels.
I was watching television with somebody, and they saturate the thing on the news show. The news shows between 7 p.m. and 9 p.m. And the person I was with said, "Why are all?" I said, "Come on, think about it. After 9 p.m., the guy watching that is just thinking about sleeping. Okay. Before 7 p.m., he's thinking about supper. So it's the prime time to influence decisions. Telemarketing. But no, the most important thing is really the response, so that the device is really cool because it even controls for the normal hiss of the pupil. So the algorithm nested in the device, you hit it with an IR pulse, the pupil changes, it records the velocity of the change, and subtracts out the effect of the hippus, which is good. We've done this reliably now in kids down to 2, because the device has a cup which really occludes light from all around the eye. And even though the kid might start out with their eye closed, they have to open it sometime. And you get an idea from that by watching the other eye. So
So
training.
obviously there's a light source in this.
Yeah, no, it produces a pulse. Yeah, it's good that way. So it's a pretty good mouse pad. It's really pretty good.
So I think we'll call the formal part of the rounds over, but please come forward and talk and ask further questions. Again, I want to thank